Welcome, everyone. My name is Shelley Bolleton, and I'm the director of the Center for Vaccine Preventable Diseases at the University of Toronto. I'm also an associate professor at both the Dalla School of Public Health and the Department of Laboratory Medicine and Pathobiology at the University of Toronto, and a scientist at Public Health Ontario. I'm pleased to welcome you to this webinar, which is entitled Human Challenge Trials Moving Vaccine Development Forward, which is co-hosted by the Center and also by the Canadian Association for Immunization, Research, Evaluation, and Education. Before we start today's webinar, I wish to acknowledge this land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. So just some housekeeping items before we begin. After the speakers each give their talk, we will try to address as many questions as we can. Please direct any questions that you have to the panelists using the Q&A function. And any questions that are not answered during the webinar can be answered if you email us at cvpd.dlsph@utoronto.ca. Closed captioning is available in English during the webinar. Instructions on how to view closed captions will be listed in the chat as well. I see that they have just now. Keep in mind it's an automated service, so we apologize in advance for any errors that are made to the captions. So now to introduce our panelists for today. So we have Dr. Christopher Chu, who is an infectious diseases physician and an immunologist. He underwent basic medical training at Cambridge and Oxford universities. His PhD was done with Charles Bingham, supported by a Wellcome Trust Clinical Research Training Fellowship. And he did postdoctoral training funded by an MRC Clinician Scientist Fellowship, during which he furthered his research by working in Rafi Ahmed's group at Emory University. Chris's research interests focus on pathogenesis and protective immunity in human respiratory viral infections, including respiratory syncytial virus, influenza, and SARS-CoV-2. To discover why some people suffer life-threatening illness, while others only have mild or asymptomatic infection, he has developed a set of unique experimental medicine techniques. He is an expert in human infection challenge studies and leads a group that uses infection and vaccination of volunteers, as well as patient-centered research to investigate systemic and mucosal immunity against these infections. These highly specialized methods that include analysis of the local responses of respiratory mucosae provide a unique opportunity to examine correlates and mechanisms of protection in disease severity in human beings. This is exemplified by his recent role as the chief investigator of the first SARS-CoV-2 human challenge study in the world, which together with his other programs aims to enhance our understanding of how respiratory viral illnesses may be better prevented and accelerate the development of more effective vaccines. Our second speaker is going to be Dr. Scott Halpern, who, in addition to being the director of the Canadian Centre for Vaccinology in Halifax, is also a professor of pediatrics and microbiology and immunology at Dalhousie University and head of pediatric infectious diseases at IWK Health Centre in Halifax. He's also co-PI for the immunization monitoring program ACTIVE or IMPACT and a member of CARE's management committee. His research focuses on the diagnosis, treatment, and prevention of pertussis and other vaccine preventable diseases. So thank you very much to both the panelists for giving us their time today. And Dr. Chu, I think you can go ahead.
Thank you, Shelley, and thank you for that kind introduction. Let me share my slides. And thank you to the organizers for the very kind introduction to invitation to speak today. So I'm going to talk to you about the SARS-CoV-2 human infection challenge work that we've been doing. And this all starts with this central question, which is really the focus for all of the work that my group does, which is what determines the outcome of infection following these respiratory viral infections. So following a virus exposure, many people completely resist infection, whereas others develop viral replication. And of these people, the majority will be asymptomatic or mildly symptomatic, with only a minority suffering life-threatening disease. So if we could understand the factors which lead people to down one path or the other, it would be possible to improve the interventions that we have. And in many ways, it's perhaps surprising that there are such great knowledge gaps in these host-derived factors. And partly that's because we've relied very much on animal models over the years, and we have extremely detailed data from mice and other small animal models. But obviously there are problems with extrapolation. At the other end of the sort of experimental spectrum, we have obviously observational studies of patients naturally infected with these viruses. But these are also extremely difficult to interpret. There are uncontrolled and uncontrollable confounders such as virus strain and dose, the environment and conditions under which people are exposed to the virus. The ascertainment of these cases is triggered almost always by symptom onset, and so there is very little analysis of early infection possible. And of course, we all understand that there are many difficulties in diagnosis and sampling, as well as the very great heterogeneity of patients with all of their comorbidities and other individualized factors, which mean that very large field trials need to be done to power the identification of statistically significant differences. So these issues have been recognized for a very long time, and so human infection challenge has been developed as a way to overcome some of these issues with confounders. So in these studies, which have, as you can see from this graph, been increasing in frequency and popularity, we deliberately inoculate volunteers with defined pathogens, and in that way control many of the confounders which I just mentioned. In respiratory viruses, the first successful study of this kind was with influenza, first conducted in 1936, and then in the middle of the 20th century, in the UK at the Common Cold Research Unit, which is pictured here on the right, but also in many centers across the US and elsewhere, whole programs of such research were really instrumental in helping us understand the effects of things like virus strain, dose, the route of delivery, transmission. You can see here a transmission study where people are playing cards with each other, and early findings about immune protection. So the key strengths of human challenge are that you can define very carefully the virus strain and dose, and all of the volunteers in any particular experiment can be given an identical challenge. And this, together with the careful selection of participants, either for their pre-existing immune status or in some limited way comorbidities, together allows the induction of very consistent and usually very high infection rates. Together with the power of longitudinal analysis, this allows small studies to be sufficient to identify statistically significant differences. And that allows us to design rapid, flexible, controlled studies, which give us the ability to generate evidence of efficacy of interventions and also the accuracy of diagnostics very early in the course of clinical development. Along with that are the ability to assess in detail the pre-infection and pre-symptomatic immune status, and in that way robustly identify protective factors which are important in the host. So in recent years, human challenge has become increasingly important as a way to accelerate vaccine research and development. Historically, this has mostly been in the early clinical phase and preclinical phase of development, where the mechanisms of protection and susceptibility have been investigated. And the best example of this probably is the hemagglutination inhibition assay, which is still used as a correlate of protection in the licensure of influenza vaccines. In our hands, we've also looked at mucosal antibodies. So here in RSV, the much stronger correlation between mucosal IgA and infection than serum neutralizing antibodies. We've looked at T cells in the respiratory tract, and here you can see just the infiltration of CD8 T cells here in green into the bronchial mucosa after infection with flu. And also importantly, although I'm not going to have much time to talk about this today, the ability to identify previously unrecognized factors which may be important in susceptibility or protection. And in this study, we identified neutrophilic inflammation as an important determinant in the susceptibility to RSV symptomatic infection. But what's really driving the uptick in human challenge work is its potential role in helping us accelerate vaccine development. And here, the idea is that large-scale efficacy trials are increasingly expensive, increasingly difficult to do, and we are still seeing large-scale phase 3 trials failing at that point, which is a huge waste of resources. And so to be able to identify candidates which are likely to succeed at this earlier phase in the clinical development pathway allows you to up and down select potential candidates and de-risk those phase 3 trials. And a good example of this is the Janssen adenovirus RSV vaccine program. This is a human challenge study which was published back at the beginning of 2021. And you can see that with only 26 and 27 people in each arm in this placebo-controlled group, there is a very clear and statistically significant difference between the placebo-treated individuals and the vaccinated individuals in terms of viral load and viral shedding. So this gave Janssen confidence to proceed to their phase 3 vaccine program, which is already looking very promising. So it's on the background of the strengths of this type of research that the WHO convened 2 working groups very early during the COVID-19 pandemic to assess the feasibility and the ethical criteria for conducting the type of research in the middle of an evolving pandemic. So there was one working group which looked at the ethics and one which looked at the feasibility. And it was building on the back of that the UK government, in the form of the UK Vaccines Task Force, decided to convene a consortium, which I then led, to try and develop this model as a way of primarily at that point mitigating the risk of first-generation vaccines failing and therefore trying to accelerate the development of potential follow-up vaccines. So if you think back to the middle of 2020, which is when we first started thinking about this, there were no vaccines. The history of vaccines against respiratory viruses is full of failure. And so there was some pessimism about the development of highly effective vaccines against SARS-CoV-2. And of course, there were no specific antivirals either. And so the rationale was quite clear at that point. The scientific and the public health benefits were very marked. And so we started on the process of manufacturing the challenge virus, which needs to be produced to an extremely high quality standard, and in this case to GMP. Alongside that, and throughout, in fact, the whole program is the potential benefit to understanding of pathogenesis and protection. But just as we were ready to submit our study protocol to ethics review, things started changing. And in particular, the first generation vaccines were shown to be highly effective and started being rolled out. In addition, treatments were being identified, and in fact, specific antivirals in the form of, at that point, monoclonal antibodies were being shown to have some efficacy in certain situations. Along with that, new and clinically significant variants were starting to emerge, firstly with the Alpha variant. And so we needed to then completely reassess the scientific and public health value of doing this type of work, which, remember, has some risks and so needs to be balanced with the risk to individual participants. So the ethical review process took quite a long time. There were multiple rounds of back and forth. And we still argued at that point that there was a need for more and better vaccines. The issues with the cold chain required for the mRNA vaccines argued for different technology platforms to become available. and the likelihood that parenteral vaccines, intramuscular vaccines, were not going to be able to fully block transmission meant that other vaccines to address that were also required. In addition, at that point, only parenteral antivirals were available, and so there was also an argument for developing the model to improve the availability of antivirals. Of course, all that since then has become even more refined, and with newer variants which are even more antigenically diverse, but on the background of boosters and very high seroprevalence and immunity rates in the community, this has become further complicated, although I think that there still is a very clear rationale now for the need for improved vaccines, given the known shortcomings of the various vaccines we have available. So to the ethics committee, their main concern was balancing the societal and scientific need with risk to individual participants. And so we really tried to distill what was unique for in SARS-CoV-2 human challenge that would generate data which was useful and could not be replicated using any other model. This started in the short term with some very significant public health questions which were still remaining, including really those early events during infection, such as the true incubation period, the usefulness of rapid antigen tests, and the impact of asymptomatic infection on onward transmission. In the medium term, we argued that the model could be used for head-to-head vaccine trials, and that's something that really can't be effectively done in the field due to differences in timing and populations. And the rapid ability to test products also involves the ability to assess the effect of vaccines and other interventions on markers of transmission. Finally, in the longer term, you can see that from the WHO vaccine tracker that there are many vaccines still in clinical and preclinical development, and it's increasingly difficult to do phase 3 clinical trials in the field, and a number of vaccines aren't going to be appropriate for immunobridging studies. And so we argued that developing this model was going to be important to provide efficacy data as phase 3 studies became unfeasible. So these are the 8 ethical principles that were set out by the WHO working group. And I'm not going to go into them all, but I'm going to focus most on risk mitigation, which is, of course, the other side of the most important balance consideration. So based on increasing field data of naturally infected people who were local to our population, over the course of 2020, we became increasingly convinced that careful participant selection would really be able to allow us to do these studies with very minimal risk to participants. And so we selected healthy people aged 18 to 30 with no significant risk factors for severe COVID-19. I've mentioned the very high quality virus that we made, and we also started with a very low dose of the inoculum, as there was a theoretical risk, and there still remains that unknown risk about higher inoculum doses potentially contributing to more severe disease. We conducted the study in a high containment quarantine unit in an NHS hospital, and had very close monitoring, as well as strict discharge criteria to minimize the risk of onward transmission. And as the various antivirals became available, we were able to bring them in so that we could implement early rescue treatment. Finally, of course, severe COVID isn't the only safety issue that is important in COVID-19, but also long COVID. And so, as well as having accumulated data which reassured us about that, we have implemented very long-term follow-up and specialist referral for any of those patients that need it, participants that need it. So this is the experimental scheme. All individuals were seronegative at screening, and they were admitted to this quarantine unit here in a London teaching hospital. All individuals were inoculated with the same strain and only 10 TCID50, which is the equivalent of 55 focus-forming units in our infectious virology assay. And the target was to achieve 50 to 70% infection rate in these individuals. Following inoculation, they were quarantined for at least 14 days, but on average ended up staying in for 16 days. And they were very closely monitored with clinical assessments, virology, and a number of exploratory readouts, including exhaled breath, environmental sampling, and a large number of immunological readouts. And we're following these individuals up for 12 months after they were inoculated. So as a testament to the public support that we had for this research, as well as the public engagement work that we did before, we found when we opened the registration website, up to almost 27,000 people who registered their interest in participating. But obviously, volunteer screening was very careful, and we whittled that down to only 36 individuals who were inoculated with this dose of 10 TCID50, which is really a very tiny dose of virus. Of these, 2 at the point of inoculation were found to have seroconverted between screening and inoculation, so they were excluded from this infection rate calculation. And in the end, 18 developed PCR-confirmed infection, which led to an infection rate of 53%. Because of this potential risk for, at least theoretical risk of increased symptoms with higher doses, and because we'd hit the target infection rate, we felt that dose escalation at this point was not desirable. Before I go on to show you some of the data from the infected people, I just wanted to quickly show you the viral load curves from the individuals who were deemed uninfected, because actually the infection criterion was 2 consecutive positive PCRs, and a number of individuals who were deemed uninfected had these individual spikes of low-level viral detection by PCR. Interestingly, none of these people had any neutralizing antibody response, and so we think that these people had abortive infections, some of them quite late down during the course of the experiment, but which was fully controlled by local or host immunity, and meant that there wasn't enough of an infection going on to induce a systemic immune response. So we're really interested in these people, and I don't think there's any other experimental setting where you could investigate these people in any detail, and we're looking into these people as we speak. So going on to the infected individuals, you can see here in the blue curves the PCR results, and in the red the culture results. And the first thing to say is that the incubation period was much shorter than we had expected. So previous estimates had been of incubation periods around 5 days, and we found that the first detection was on average at 40 hours, and that was first in the throat and then later in the nose. But in nose, the viral load was much higher, and really huge viral loads were found, so 10 to the 7, 10 to the 8, and in some people up to 10 to the 10 copies per mil. The shape of these viral load curves was very consistent between individuals, but the timing could be quite variable. And that has an impact on public health messaging, and we were feeding these data directly into government committees in the UK to help decide on some of the public health decision making. On average, individuals were shedding infectious virus for about 6.5 days after virus exposure. But you can see that some individuals had detectable infectious virus up to day 12 after exposure, and some cleared very quickly after only 2.5 days. Happily, all the individuals were quite well over the course of the infection, and in general had very mild upper respiratory tract and systemic symptoms, which is expressed here in the red curve as a sum total symptom score, which peaked around day 4 to day 7, day 10. Individuals who were not deemed infected had no symptoms at all. But almost all the infected people also had quite significant changes in their sense of smell and taste. And this was measured using the University of Pennsylvania smell identification test, which is here shown in purple. So in about 80% of the infected individuals, some measurable smell disturbance was found. This peaked later than other symptoms, probably about a week later. And in most people, resolved very quickly with complete resolution by day 28. A few people did have more protracted symptoms of reduced smell sensitivity. And in particular, one participant at day 180 still had some measurable smell reduction, although this was relatively mild subjectively. And the literature does suggest that by 12 months, almost everybody completely goes back to normal. So we're awaiting further improvements. Importantly, while the symptoms correlated with viral load in terms of timing, they don't correlate with viral load in terms of magnitude. So you can see that even the most, the people with no symptoms at all were still shedding very high levels of virus from their throat and their nose, which really explains why there has been so much ongoing transmission despite relative reduction in symptomatic disease. Finally, in this first study, what we wanted to look at was how the rapid antigen testing how effective that was or how accurate that was in the early part of infection. And in the UK, lateral flow rapid antigen tests were implemented very early as a key part of the public health response. But actually, our understanding of the early events during infection and how accurate lateral flow tests are during this time were really lacking. And what you can see in the black bars is the sensitivity against qPCR or against infectious virus. You can see that lateral flow tests are only around 50% or less sensitive at the beginning of the infection, the first 2 days of viral detection, but become very accurate during the main part of infection, tailing off again as the viral load decreases. But importantly, the sensitivity remains high throughout, and particularly at this latter stage where we would be using lateral flow to enable test to release and to shorten the duration of self-isolation. This is sort of aggregated in this analysis in collaboration with Neil Ferguson, which showed that if you use the recommended lateral flow testing strategy that was implemented in the UK, where people were recommended to test themselves every 2 to 3 days, you would capture around 80% of virus. And if you implement self-isolation, you would be able to interrupt transmission of potentially 80%. So I think the key learnings that we had from this study, which I'm going to conclude with, are that this study couldn't have been done at all if pre-existing networks and relationships hadn't existed. So in the UK, we had a network called HICVAC, which was funded by the Medical Research Council, and also a number of inter-institutional vaccine networks, which built up those relationships and had us ready to be able to get going on a study like this, where clearly there were some controversies and required a lot of thought by experts and also people involved in public and governmental response. I think that these studies are going to give us a huge amount of additional data, which as we study those samples which we've collected. But all of this has been necessary it's been necessary to consider in the context of a very rapidly changing pandemic landscape, which has brought all sorts of challenges, which I've tried to express in some of the earlier slides. Going forward, what we're particularly interested in is why those 47% of participants resisted infection. And we anticipate through our in-depth analysis of transcriptomics and immune response that we will be able to identify new targets for developing preventive strategies as new pathogens emerge. But obviously, in the UK, certainly, a large majority of individuals are now positive for antibodies with immunity either from previous infection or vaccination. And so this seronegative study is unlikely to be replicated ever again. Instead, we're moving on to a new model of breakthrough infection with viral variants. And so we've manufactured a GMP Delta challenge agent, and we are hopefully going to shortly start a Delta challenge study in vaccinated and previously infected individuals to understand the determinants of breakthrough infection and optimize a platform for testing of future interventions. In particular, for vaccines, I still think that improved vaccination against SARS-CoV-2 is required. We need transmission blocking vaccines, and we need vaccines which confer cross-strain protection that don't require multiple boosters, and certainly annual boosters. And for many of these vaccines which hope to achieve these aims, I believe that unconventional approaches which don't induce serum neutralizing antibodies or high levels of serum neutralizing antibodies are going to be required. And so immunobridging studies are not going to be appropriate for those, particularly things like mucosal vaccines or T-cell inducing vaccines. So I see SARS-CoV-2 human challenge as being able to provide rapid small-scale studies with efficacy readouts, including reduction in viral load as a surrogate for transmission, and provide proof of concept against diverse strains, including Delta and the Omicron virus, which we're hoping to manufacture in the near future, without necessarily having to chase contemporary variants sort of as they come online. So I'd just like to thank all of the people involved. This has been a huge endeavor of work and required a lot of collaboration between many different institutions and individuals. And I'd like to, again, thank the organizers for the opportunity to speak.
Thank you, Dr. Chu. Over to you, Dr. Halpern.
Thank you. Can you see my slides?
Not quite yet.
Okay. Let me know if you can. It says it's sharing. I'll try again.
Yep, we see them.
And do you see them in the full screen?
Now we do, yes.
Okay, great. Sorry for the delay. Thank you for inviting me to talk. And there are a number of my slides which overlap, so I will just skip them as we come to them, just to save some time for questions. So first, my disclosures for research funding and consultancies. Many of them related to pertussis, which I'll be talking about. So what's the burden of pertussis? Why is this still an issue? I mean, SARS-CoV-2, that's novel. Pertussis, we've known about for hundreds of years. So classically, pertussis was a disease of infants and children, peak incidence in preschool age children with most mortality in young infants. And by school entry, about 50% of children would have had pertussis. In the current vaccine era, pertussis affects all ages. It still peaks in young infants, but also in older children and adolescents. But morbidity and mortality remain in young infants. what we're seeing is a reemergence of pertussis in Canada and elsewhere. And if you look at the red bars, you can see that the incidence is starting to rise again. Still not quite up to, in Canada, to the pre-vaccine era, but in some areas, in the US, for example, they've had some years that rival the pre-vaccine era. So why is pertussis reemerging? And it's probably multifactorial. The pathogen may be changing. There are now protectin negative strains. There are strains with altered pertussis toxin excretion. Pertussis is a cyclical disease, and some of it is just there are now larger peaks in the typical 3 to 5 year cycles. There's also more awareness and reporting. There's improved surveillance, better diagnostics. And we still have the problem with some failure to vaccinate. But vaccine failure is also an issue, and where I'll be focusing most of my talk. Pertussis is a difficult illness to diagnose. It's not suspected typically until the paroxysmal phase, when microbiologic diagnosis is no longer possible. It does have the cyclical epidemiology, so it's difficult to study because you don't know where it's going to be in a particular year. The mechanisms of immunity and correlates of protection are still not known, and we don't know a whole lot about the transmission dynamics, carrier states, mild infections, and the duration of protection is also different between natural infection and post-vaccine. And again, the efficacy of vaccines, which is where challenge studies may be a real benefit. The current vaccine, the acellular pertussis vaccine, compared to the whole cell vaccine, has advantages, and it's better tolerated because of lower adverse events. The acellular vaccine is easier to standardize production, but it may be less effective than the best whole cell vaccines. And this is primarily because of its Th2 biased immune response, which may lead to a short or shorter duration of protection. In this slide, what can see in an outbreak, the incidence of PCR positive cases were the lowest in individuals who had had an all whole cell primed immunization series. If they had a mixed series between whole cell and acellular, the incidence of PCR positivity was in the middle, and those who had all acellular pedigree in terms of their vaccination status had the highest rates of PCR positivity. And in some data from Canada, one can see that as increasing interval between since the last immunization, you can see that the positive tests increase, so that the pertussis vaccine in this group was reasonably effective for the first 2 to 3 years, but then started dropping quite dramatically, so that by 9 to 10 years, there was really little protection left from vaccination. So new pertussis vaccines are needed, but it's a costly endeavor. One needs to understand better the dynamics of transmission and immunity. And the regulatory pathway is quite difficult because pertussis vaccines are used widely around the world. So a placebo-controlled trial is impossible to do now. And most of the advances over the past 20 years have been to bridging with the initial acellular pertussis vaccine efficacy studies, which were done in the 1990s. And there's no longer ability to bridge to those studies because all the reagents are now gone. A baboon model of pertussis has been developed, and most work had been done in animals and mice were good for studying a number of things, but don't mimic the disease very well. Todd Merkel and his group at the FDA, Todd's the lower person, not the upper, he developed a baboon model which had more of the classical symptoms with a paroxysmal cough, and it's been very useful for studying transmission and infection. So for example, in the baboon model, the whole cell, if one's looking at disease, the whole cell pertussis vaccine prevents it, as does the acellular vaccine. But for colonization, the whole cell pertussis vaccine prevents colonization where the acellular doesn't. And the whole cellular vaccine prevents transmission and the acellular doesn't. So these are some potential reasons why the vaccines are failing and we're seeing increased cases of pertussis. So a controlled human infection model, or a CHIM, would be a good way of studying some of these things to ensure that the baboon model, and to expand on the findings from the baboon model in humans. As Dr. Chu mentioned, challenge studies are very useful for establishing both transmission and understanding pathogenesis. So I won't belabor these points. The challenge research can potentially look at a vaccine efficacy and contribute to understanding the various markers, particularly correlates of protection, which would then be able to allow the licensure of new vaccines based on immune correlates rather than on field efficacy studies. So as Dr. Chu mentioned, vaccine challenges allow you in a quick and cost-effective way of looking at efficacy at the early stages of vaccine development. It allows you to make formulation changes. It can allow you to look at carriage, transmission, the severity of infection. So despite the fact that pertussis is a bacterial infection, the types of things we can find from vaccine challenge are very similar to what was talked about with SARS-CoV-2. Pertussis meets the criteria for principles of pathogen selection, is that the pathogen is well characterized, the disease is self-limited in healthy individuals, and that there's a rescue therapy allowed to prevent the establishment and the occurrence of the complete whooping cough syndrome. Importantly, though, the pathogen has to be approved by either the FDA or Health Canada or the MEA for use in humans, so that the strains now have to be go through the full process as if it was that you're developing a new drug or a vaccine. You also need to have concern about infection control and prevention and control. So biologic containment is important. What are the risks to people outside of the institution, and what can you do if any contacts of your participants develop symptoms? So there are a number of things that argued for the development of a pertussis CHIM in order to understand a variety of pathogenesis issues and issues around diagnostic tests, chemoprophylaxis, and as a potential regulatory pathway for new vaccines. So should a human challenge model of pertussis be developed? Well, in fact, it already has, and a group in the UK and in Europe, as part of the Periscope group, has developed a CHIM model, which had their first challenge just prior to the pandemic, and have published on their results already. So why do we need an additional challenge here in Canada or in North America? Well, there are a number of advantages that one can have. One can study additional endpoints or outcomes. One can look at different challenge strains, different immunologic pedigree of the participants, increase capacity in order to do the studies, because you still need if you're going to use it for licensing a vaccine, the studies still have to be a little bit larger, than just what you need for looking at some of the pathogenesis aspects. So as I mentioned, the current vaccine provides suboptimal protection. The animal models are insufficient. The Periscope study has developed a model that's very effective. And a CHIM in the context of North America, who's had a different immunization schedule, would be beneficial for licensing vaccines in this context. So what the reasons why we're developing one here in Canada is for additional outcomes. The Periscope model looked at colonization as their outcome. We'll be developing the model to both look at colonization and early disease. So going up to include the catarrhal stage of pertussis, but then beginning therapy before people go into the paroxysmal stage. We will be using a North American strain, the D420 strain, which is the strain that was used in the baboon model, in order to correlate a lot of what we find in the human model with what was been seen in the baboon, and to add to that growing body of evidence. The differences in the schedule in the UK and in Europe. Acellular vaccines were introduced later. There's different primary series using 2 doses rather than 3. And in North America, we use 5 doses before school, as well as adolescent booster, which is far more than is used in the European context. So this is the detailed study of what our model is, our CHIM study is, and Maël Sharif at the Canadian Centre for Vaccinology is our lead investigator who's moving this forward. I won't go through the protocol in detail because I want to talk more in the next 5 or so minutes about the process of getting the CHIM model approved. But our objective in our first study is just a dose finding study, followed by a dose confirmatory study. And the main objective is really to look at the development of colonization in early clinical illness. We'll also have a number of exploratory objectives in terms of the timing, best timing for both looking at the immune correlates, both humoral and T-cell immunity. Our process is we'll have a screening period followed by a challenge and then an inpatient in an isolation facility for 17 to 21 days, followed by outpatient follow-up visits that last a year. So the challenges to developing a pertussis challenge, and for any challenge, is first funding. No one wants to fund the development of model. Everyone wants to fund using the model. That became a real blocking point. And the way the CHIM model was developed in Europe was a consortium of federally funded, a consortium across Europe to particularly develop the model and then utilize it. For us, we had to use internal funding to do the biomanufacturing. We were able to get an NIH grant to develop the protocol and study related documents. And ultimately, the CDC, through one of their broad agency announcements funding mechanisms, funded the establishment of the dose finding and dose confirmatory study. With the CDC's main goal is to have a model which could then be used for a regulatory pathway. So very similar to the success of the SARS-CoV-2 challenge, which you've heard about, was that government policy to say that this was an important mechanism to get important answers and to take a lead in the funding of it and organizing that consortium. The regulatory process, as with any other clinical research with humans, the challenge study has to be conducted according to ICH guidelines of good clinical practice. The strain to be used must be selected, well characterized, and increasingly manufactured according to GMP. We were fortunate to allow the regulatory authorities, because it's a bacteria and can be grown in cell-free conditions, we were able to use GMP-like methodology rather than GMP, which was a huge cost savings and allowed us to manufacture the strain ourselves. A lot of the work was aligning the comments from the 2 regulators because the CDC wanted it regulated by the FDA as well as Health Canada, where the study was going to be done. As I mentioned, biomanufacturing was critical. The GMP-like manufacture was a great benefit, but it was an iterative process. And fortunately, Health Canada was very helpful in this, in that as we developed the methodology, we were able to meet with them and get feedback on what they would require to approve the strain, so that ultimately when it was submitted, it went through very quickly. And finally, research ethics. It was necessary to establish a comfort level with the local REB. And we did this by meeting, unrelated to the specific protocol, first with the REB to discuss the history of CHIMs, the ethical principles, recruitment, compensation of volunteers, rescue therapy, physical space, and the SOPs. So before we even submitted anything, we had a discussion, a detailed discussion, so that when the actual protocol was submitted, it went through quite smoothly. And we as investigators were able to meet with the REB before they deliberated on the specific protocol, and this allowed the protocol to be approved quite efficiently. So the current status of our CHIM is that Health Canada approval has been received, the FDA approval has been received, our REB approval has now been received, and recruitment began a couple of days ago with our anticipation of the first participants being challenged by the 3rd or 4th week of this month. This is our challenge unit, which is a 10-bed full containment unit built specifically for vaccine research at the IWK in Halifax. There's a sister unit at McGill in Montreal, which we anticipate after we've established the model, we'll then have a total of 18 beds to be able to do larger challenge studies. And I'll end there. Thank you.
Thank you very much, Chris and Scott, for those very fascinating presentations, and very interesting to hear about your experiences and some of the similarities and the differences. So I just want to encourage people to put questions in the Q&A, and we have about 5 to 10 minutes or so for questions. Before we go into that, I'm going to do a shameless plug for CARE. So as mentioned at the beginning by Shelley, this is co-developed with the Centre for Vaccine Preventable Diseases and CARE, which is the Canadian Association for Immunisation Research, Evaluation and Education. And I've just got a couple of quick slides. So for people who are not aware, so CARE is a professional organisation with a mission to enhance immunisation research and programme evaluation across Canada through education and collaboration with multidisciplinary experts. And there's the Twitter handle and the email address. We're just launching a slightly different model of membership to CARE, which will involve a relatively nominal membership fee, and this will give people access to an extensive network of professionals supporting vaccine research and immunization program development, evaluation, and training, opportunities for CARE funding and awards, which usually seems to be the carrot that brings most people in, but also supporting co-developing workshops, panels, other educational opportunities, networking at various events, and various other members only web resources, which are some are developed, some are in development. There's a QR code there. The website is almost finalized and ready to go. And I think Beth is going to put a link in the chat, or someone's going to put a link in the chat as well, that will take you there. And just a quick plug for a couple of other upcoming webinars. So we have a co-developed webinar with CanVax on how immunization research informs policy decisions, and then another one next month with CanCovid on immunization research with community partners, and some excellent speakers to come. So hopefully that has given you a bit of a flavor for CARE, and also some time to ask questions. And so I'm going to get started here with some questions that have come in. So Chris, this first question is specifically for you. So maybe you could speak a little bit about why the next challenges will be with Delta as opposed to Omicron. And can you, I think you did mention it, but can you just confirm which variant the strain was based on for the data you presented already?
Yeah, so strain development is really a bottleneck in this kind of research, because I mean, it's true for all challenge agents. It's absolutely critical that they're manufactured to high quality and that they're extensively tested, and all of that means that it takes quite a long time and a lot of cost to manufacture a stock of a challenge agent. So in our first study, we obtained the swab in the summer of 2020, and so it was a pre-alpha D614G containing Wuhan-like strain, and it took almost 9 months to get that fully released. Partly that was because there were not many sites which had containment level 3 GMP manufacturing capability, and there was a lot of competition for that sort of lab space, and so it was not only very expensive to do, it was also very delayed, waiting for slots and so on. So the reason we didn't go for Omicron is that at the next opportunity that we could manufacture, Delta was the prevailing strain. And so we just went ahead with Delta. I mean, there was no way to predict that Omicron was on the horizon. You can argue that Delta is antigenically diverse, not as antigenically diverse as Omicron, obviously, but it also causes breakthrough infection. And so I think it was a reasonable virus strain to go with. Just bear in mind that the cost of manufacturing a containment level 3 pathogen to GMP is about £2 million, which is about 3.5 million Canadian dollars, I guess. So it's not trivial. There are funders who are interested in it. You know, they're strongly in favor of or see the value of the model, and so we are working with them to see if we can put together funding for an Omicron manufacture.
Yeah, no, I think very difficult to keep up with anything, whether you're doing human challenge or not, with the pandemic and the progress.
Yeah, and just to say that I think that chasing after variants is kind of fruitless because it's unpredictable, and with all those costs, you can't just sort of keep chasing your tail. So the way that I see it is that we are developing tools which are antigenically divergent, and that they can together be used to generate proof of principle for cross-strain protection, and that's as good as we should be, as good as it can get, really.
Thanks. So Scott, so one for you. So there's a question, well, I guess specifically to pertussis, but we can expand it a little bit. You know, it's mostly a pediatric disease. So where do you see any future for human infection challenge models in children for pertussis or for anything else? And in general, can you speak a little bit about where you see the future of human challenge studies in Canada?
Sure. I don't think it's possible to do CHIM studies in children. I think most ethics boards would not be satisfied with parental authorization for a challenge. They would require consent. So you might be able to do it in older adolescents who can give consent for themselves, but I don't think parental authorization would be deemed sufficient for a controlled human infection model. At least not in the current context. I mean, I think if one became very comfortable with these and there were a lot of data, one might be able to go further in if they were shown that there were no individuals that needed symptomatic rescue therapy, but just rescue therapy based on we're terminating the infection. Then in the future that might be possible, but I would doubt it. And mostly, you know, the vaccines are first going to be most needed in infants, and there's no way that any ethics board would approve a CHIM in an infant. Where I think they will go is looking at developing vaccines that can potentially overcome that Th2 bias. I think there's a lot of hesitancy about interfering with the infant immunization schedule, because if you change the pertussis vaccine, you have to change the combination vaccines with, you know, that because they're combined with tetanus, polio, Haemophilus, diphtheria. It's a lot of work to change all those. So what one hopes is that one could, you know, have a booster that develop a booster that has longer duration and improves the current vaccine in terms of duration without having to change the whole model. Now, having said that, there are a company that's it came from the Pasteur Institute initially, who has a live attenuated pertussis vaccine that's undergone clinical trials, and that will be tested in human challenge models, and that potentially could be given to infants, you know, even prior to their first dose, either as a primary series or prior to the primary series, in order to set the Th1 bias prior to using the alum adjuvanted current acellular vaccines. So I think that model is really going to help understand the immunology better, and then be used as a licensing pathway for new improved vaccines.
We have sort of reached the end of our time. Unfortunately, I think we could keep talking about this for a long time yet. I just want to thank both of you for sharing your time and your experiences today. I think it's really exciting to see the data coming out of these models, not only for SARS-CoV-2 and pertussis, but I think a lot of other infections, as you mentioned, Chris, in your graph. This seems to be a really expanding area for sort of infectious diseases and vaccines. Just a couple of concluding remarks. So the webinar will be posted on the CVPD website, and there's a link there in the chat. If people who have questions that are not answered, please do email them to the cvpd.dlsph@utoronto.ca, and again, that is also in the chat, and then we'll do our best to get them answered for you. And as I said, please do join up for membership of CARE and also for the CVPD mailing list as well. And there's a link there as well. So lots of links for you. And I just want a huge thanks to Beth White from CARE and to the CVPD team and to Shelley and Shaza for helping support and bringing this webinar together. So thank you all very much and enjoy the rest of your day.